All right, this presentation goes along with the Lab 3A Understanding pH and Buffers PowerPoint slides that are available on Blackboard under Lab 3. You should also watch this video before you do the Lab 3A Prelab. Now, we talked about pH and buffers a little bit in class. We talked about what an acid is and what a base is. If you're still having difficulty with those concepts, then I recommend that you go to the Lab 3 folder on Blackboard and that you find the animated modules that I've posted there. There's several really great animated modules that take you inside a solution and show you what it looks like, what's going on in there when there's an acid present and when there's a base present. So I would recommend that you check those out. Now, we've got several important concepts to cover in this lecture. In this lab, what you're going to do is you're going to make several different solutions. You're going to make unbuffered solutions and buffered solutions. You're going to add acid to the solutions, and you're going to add base to the solutions, and you're going to see what happens to the pH as you do so. So you need to understand what's going on inside that solution. So the first thing we're going to talk about is concentration of acid and base, what it means, how they relate to one another, and how that relates to the pH scale that we discussed in class. So first, you need to understand this. If you had a sample of very pure water, just H2O, and you were to look inside that pure water to see how much acid was present and how much base was present, what you'd find is that collectively, there's about 10 to the negative 14 molar of these substances present. Now, what does that number mean, 10 to the negative 14 molar? Well, if you write that out instead of using an exponent, you get this number. You can see that it is a very, very, very tiny amount of acid and base collectively in pure water. Now, because of this estimation, 10 to the negative 14 molar, 14 is going to be our magic number for this unit. So keep 14 in mind. Now, another thing you need to understand is that the concentration of hydrogen, which as we discussed in class is an acid, and the concentration of hydroxide, OH negative, which as we discussed is a base, is alkaline. The concentrations of these two chemicals are going to be inversely related to one another in a given solution. Now, the concentration of a chemical is shown by these brackets. So this bracket that says, a bracket with H plus in it, that's saying concentration of hydrogen, and this is saying concentration of hydroxide. So that's what those mean. But let's say that I am told that the concentration of hydrogen in a solution is equal to 10 to the negative 5th molar. Well, using this information, I can figure out what the concentration of hydroxide is in that solution. And the reason is that the exponents on these concentrations are always going to add up to 14 because of this estimation that I gave you before, 10 to the negative 14. So if I am told that the concentration of hydrogen is 10 to the negative 5th, I know automatically that the hydroxide in the solution is going to be 10 to the negative 9 molar. Now, how did I know that? Well, 5 plus 9 is 14. And the exponents will always add up to 14. Okay, given that, I would like you guys to pause the video at this point and try these two problems. I want you to take a concentration of hydrogen and figure out the hydroxide concentration. And I want you to do the reverse with the hydroxide concentration. Okay, so pause it, and then we'll look at your answers. Okay, let's take a look at your answers. In this first problem, you're told that the hydrogen concentration is 10 to the negative 3, and that should be molar right there. And you're asked what the hydroxide concentration is. Well, since our magic number is 14, 14 minus 3 is going to be 11. So my concentration of hydroxide is 1 times 10 to the negative 11 molar, or I could just say 10 to the negative 11 molar. In this problem, you're told the hydroxide concentration is 10 to the negative 13. Since 13 plus 1 is 14, that tells me that the hydrogen concentration must be 1 times 10 to the negative 14, or sorry, the negative 1 molar. All right. Now the next thing we need to discuss are logarithms and pH and what all of this has to do with concentration. It's actually pretty easy to flip back and forth between the concentration of hydrogen and the pH, and I'll show you how it's done. So first of all, let's look at the calculation for pH, how they determine the pH of a solution. The pH of a solution is based on the hydrogen concentration. They could have based it on hydroxide, but scientists decided to go with the hydrogen concentration. Now, the hydrogen concentrations, as we noted in the previous slide, are these very, very, very small amounts, 10 to the negative 9, for instance. And that number can be a little bit difficult to wrap your head around. So what's been done here is that scientists took that estimation, 10 to the negative 9, molar, and they took the negative log base 10 of that. Now, why would you want to do that? Well, it turns out that if you put this in a logarithmic form, this number like 1 times 10 to the negative 4, it comes out as a number that's easy to understand, 4. So the fact that they used logarithms to calculate pH is actually really handy for us. Now, what is a logarithm? A logarithm is a way of asking what exponent you can raise a number to get a second number. For instance, if you have the log base 10, what you're asking is, what number can I raise 10 to? What exponent can I use to get this number? 1,000 in this case. So if you're asking, what number can I raise 10 to get 1,000? The answer is going to be 3, because 10 to the third is 1,000. Okay, so that's why this pH calculation works. If you take this number 1 times 10 to the negative 4 molar and you put it into this equation, what it does is it puts it into a form that's easy to understand, 4, and it also gets rid of that negative sign that's on the exponent. Okay? So it makes it easy to understand. All right, now here's the trick with switching between concentration and pH. As it says here, the exponent of the concentration is equal to the pH without the negative sign. So, for example, if you are told that the hydrogen concentration of a solution is 1 times 10 to the negative 4 molar, and you're asked for the pH, all you have to do is take that exponent and ditch the negative sign, and you've got your answer. Since this is 10 to the negative 4, your pH is 4. And it really is that simple. If you're told the pH is 4 and you're asked for the concentration, you just use that 4 as a negative exponent on 10, and you've got your concentration in molarity. All right. This slide is showing that same thing. Here's another example over here. Again, if your hydrogen concentration is 1 times 10 to the negative 6, and you are asked for the pH, pH is 6. Take that exponent, you ditch the negative sign, you got your pH. Okay. So another way to think of this is that the hydrogen concentration is equal to 10 to the negative pH. That pH is going to be your exponent. All right. Now, so far we've looked at pHs that are nice whole numbers like 4 and 6. When you actually use a pH meter during lab, it's probably not going to crank out nice whole numbers for you. For instance, it might tell you that the pH of a solution is 3.2. Now, if you're given that pH and you're asked the hydrogen concentration, what are you going to do? Well, it makes sense in context with what we've been talking about. that you would make that pH number your negative exponent. So you might say the concentration is equal to 10 to the negative 3.2 molar. And technically that's correct. But in mathematics, it's considered bad form to have an exponent with a decimal place. So if you're asked this, what you want to do is take this number, 10 to the negative 3.2. You want to put it in your calculator and press go. And it's going to crank out another number for you. So if you put 10 to the negative 3.2 in your calculator, it's going to tell you that that is equal to 6.3 times 10 to the negative 4 molar. And that's true. Okay? Mathematically, that's true. But this puts it in a form where it is in scientific notation, but your exponent that's being used here does not have a decimal place. So that's more acceptable. Okay, so that's something you might run into. All right, so we were talking about hydrogen concentration. Let's see if we can get the hydroxide concentration from the pH. It's also really easy. All right, I want you to think of this. Since hydrogen and hydroxide are inversely related to one another, as we discussed before, what that means is that the concentration of hydroxide is going to be equal to 10 to the, not negative pH, but 14 minus pH. Remember, 14 is our magic number. So if you subtract the pH from 14, that'll give you that negative exponent that you need. So for instance, if you are told that the pH is 4, then the hydroxide concentration is going to be 10 to the negative 10 molar, because 10 plus 4 is 14. If you are told that the hydroxide concentration is 1 times 10 to the negative 6 molar, that tells you that your pH is 8, because 8 plus 6 is 14. Okay, so the only extra step you have to do with hydroxide is remembering to subtract that number from 14. And the conversions are just about as simple. All right, at this point, I'd like you to pause the video again and try these conversions and see how you do. And I will check back in with you in a second. All right, hopefully you had a chance to try those problems. Let's see how you did. This first one, pretty easy. You're told the hydrogen concentration is 10 to the negative 3. You take that 3, you ditch the negative sign, your pH is 3. In the second one, you're told that the hydroxide concentration is 10 to the negative 10. That means that the pH is going to be 4, because 10 plus 4 is 14. You're told the pH is 6. What's the hydrogen concentration? Easy peasy, it's 10 to the negative 6. Or technically 1 times 10 to the negative 6. Here you're told the pH is 9, and you're asked for the hydroxide concentration. What you want to do is subtract that 9 from 14, and that's going to be your negative exponent. So 1 times 10 to the negative 5 molar is equivalent to a pH of 9 in terms of hydroxide concentration. All right, so that's the acid and bases part of this lab. The other thing you're going to be working with in lab 3 are buffers. Now, buffers are solutions that help stabilize pH. They are resistant to changes in pH. So in other words, if you add a buffer to a solution and then you add some acid to it, it's less likely to have a pH change. If you add base to it, it's less likely to have a pH change. So what you're going to do is you're actually going to make 3 solutions in lab 3. You're going to make a sucrose solution that's unbuffered. You're going to add some acid to it. You're going to add some base to it. You see what happens. You're also going to make a sucrose solution with an acetate buffer. You're going to add acid and base to it and see what happens. And you're going to do the same thing with a third solution with a bicarbonate buffer. Now, once you're done, you're going to compare the characteristics of your two buffered solutions. And you'll compare it with your unbuffered solution too. I think you'll see a big difference. Two things you can analyze with buffered solutions are the buffering range and the buffering capacity. So let's take a look at range first. The range for a buffered solution is the pH at which the solution resists change. So we're looking for pH. Let me show you an example of the kind of thing you're going to be looking at. This graph summarizes essentially the experiment you're going to do. What they did here was they took a solution and its initial pH was around 10. And then they started adding microliters of HCl, of acid, to it. And they kept track of the changes in pH. Now what you can see happening is that it starts out at about 10. They add a little bit of acid, about 100 microliters, and the pH drops. So it was not resisting change there. But at about pH of 8, it starts to stabilize. It's changing less rapidly, even though they're putting quite a bit of acid in the solution. Once it gets to a pH of about 7, you see another drop. So it stopped resisting the change. And then it stabilizes again at around 4. So for this particular buffer, you could say it has two ranges. It has two pHs at which it best resists change. And that would be from about 8 to about 7. And again, from about 4 to about 3, thereabouts. Now, the other characteristic you're going to analyze with your buffers is the capacity. The capacity has to do with the amount of acid or the amount of base you had to add to that solution to get that pH to change. Let's take a look at that. Now, I won't give you a chart representing a buffered solution and say, what's the capacity? But what I might do is give you charts for two different solutions and ask you which one has the greatest capacity. So let's take a look at these two graphs and see what's going on in terms of capacity. This is what your graphs are going to look like. This is called a titration curve. So this group, they started with a solution that had a pH of about 5.5-ish, and they began to add acid to it. So 1 mil, 2 mil, 3 mil, and so forth. They added 5 mils of acid before the pH began to drop. So this solution was able to absorb 5 mils without changing. They also added base to it, and look what happened there. They were able to add about 4 mils of base before the pH started to change. So you've got a range of about 9 mils of chemical that were added to this solution without tremendous change. That's a pretty big capacity, comparatively. Now let's look at solution B. Look what happened here. They started out with a pH of around 7. They added 2 mils of acid, and then they got a big drop. They then got another little stabilization for about 2 more mils, and then they got a big drop. Look what happened when they added base to this solution. They started adding base, and it went right on up. There were no points of stabilization, no buffering ranges for the alkaline side of this chart. So when you compare these two buffers, what you can say is that solution A has a bigger capacity than B, because they were able to pour a lot more acid and base into A, and it resisted that change, as compared to B, which changed a lot. All right, now the last thing you're going to need to do for this lab is to prepare the buffered solutions. Seems really complicated, but it's actually not. So let me tell you how to do that. Well, in lab 2, you were preparing solutions, and essentially all of your solutions had 2 ingredients: KMnO4, potassium permanganate, and water. Here you're being asked to prepare a solution with several ingredients. For instance, in this problem, you're asked to make 100 mils of solution, and it needs to contain all of these ingredients with all these final concentrations. Seems overwhelming. How do you do it? Well, essentially what you're going to do is you're going to do a series of parallel dilutions. You're told, for instance, that you have this stock concentration of sucrose and that your final concentration needs to be this. So what you can do for the sucrose is you can use the good old C1V1 equals C2V2 formula on this ingredient, and you can figure out how much sucrose you need to add. You're going to do that separately for these two other ingredients. So you're going to make 3 different calculations to figure out how much of each ingredient to add. Now, you've done these calculations before, so they're pretty simple. This just shows mathematically what's going on. So for sucrose, you're going to fill in your stock concentration for C1. You don't know your V1. You're going to set that equal to the final concentration you want to make times the amount you want to make. Oop, no. There we go. The amount you want to make. And then you solve for V1. In this case, it tells you you want to use 20 mils of that stock. You do the same thing for the other 2 ingredients. Now, the last ingredient is going to be water. So how much water do you want to add? Well, all you have to do is add the volumes of the other ingredients together. 30 plus 20 plus 20 gives you 70 mils. And you subtract that from the final volume you want to get. So in this case, you wanted to make a 100 mil solution. You subtract those volumes from 100 mils. That tells you want to add 30 mils of water. There's an even easier way of making that solution with the proper amount of water. And here it is. Once you know the volumes of the various ingredients you're going to use, you can just combine those in a graduated cylinder and then just add water to it until it comes up to the final volume that you want. And that'll be 30 mils anyway, if you're measuring correctly. All right, so that's how you make a buffered solution with multiple ingredients, and you guys are going to practice that and make those solutions in lab. Really not hard, just takes a little practice. All right, at this point, you can now go back and complete the pre-lab for lab 3A, having to do with acids and bases. Lab 3B, which is about microscope, we will likely do the following week, so you don't have to worry about it this week, unless otherwise instructed. All right, I hope this helps. Email me with any questions, and thank you very much for your time.